Good afternoon, everyone. My name is Hakon Hogset, and as James said, I just finished my PhD with Molly Stevens at the Imperial College. And in this talk today, I will present work from my PhD, developing confocal Raman spectroscopic imaging for biomolecular analysis in zebrafish embryos. And I will try to highlight some of the advantages of this methodology, namely that you can perform a label-free analysis and simultaneously access a wide array of different biomolecules and do this analysis in living and intact biological specimen. And I will demonstrate two applications that we developed in this work, where one is a three-dimensional biomolecular discrimination of infecting bacteria inside fish, and also time-lapse imaging of the wound response in living zebrafish embryos. So I will first start by introducing Raman spectroscopic imaging for anyone who might be unfamiliar with the method. So Raman imaging makes use of the Raman effect, which is caused by inelastic scattering of photons when they interact with chemical bonds. So what we do is we shine monochromatic laser onto a sample, and a small proportion of the photons will then be Raman scattered and give rise to a wavelength shift. So these wavelengths are specific for the molecular bonds causing them, and this means that we can generate this spectra containing chemical information. And because many of the common biomolecules, such as proteins and lipids and nucleic acids, have Raman active chemical bonds, we can collect these spectra to get a description of the biomolecular composition of our samples. So then, combined with point scanning lasers, we can generate biomolecular maps of the samples, where each pixel is basically one of these spectra that I just showed you. And this has been a focus in the Stevens group for the last decade. Here is an example of what a Raman image can look like, and this is obtained from cartilage tissue and was published in 2016. So why would this be interesting in zebrafish embryos? Well, mainly zebrafish has some fairly interesting characteristics as a model. So the zebrafish embryo, which you can see here, is optically transparent, which makes it ideal for visualizing biological processes. It also comes with numerous disease models, including cancer and bacterial infection models. There are currently more than 240 different bacterial infection models in the zebrafish. And it's fairly easy to manipulate genetically, so it's a good model to work out mechanisms, and has been used quite a lot, both in developmental biology and in disease modeling, to work out mechanisms genetically as well as on a cellular level. So the key motivation for us then was to kind of make use of this model to perform biomolecular analysis of disease progression and development in an intact biological specimen. And we actually started first by acquiring 3D images of whole zebrafish to basically see if this method could work. And what we observed was that we could obtain really high quality Raman spectra from most of the embryos, which you can see here is a stack covering 100 micrometers. We saw a bit of scattering in the head region and in the yolk sac region, which I'm pointing to here, but otherwise from the rest of the embryos we got really nice signals. We also observed a bit of pigment, especially on the surface of the fish. But deeper into the tissue we saw less of the pigment. So we moved on from this to acquire high resolution images of zebrafish tissue, and this enabled resolution of really fine anatomical features. You can see two examples here. We have muscle tissue to the left and tail tissue to the right, and we can clearly see bulk collagen associated with the muscles, also cell membranes and DNA from individual cells. The same in the tail, you can clearly see the DNA corresponding to cells as well as collagen fibers in the tail. So this demonstrates the quality of the biomolecular information we could obtain using this approach and enabled us to move forward. And we were next interested in kind of using this confocal Raman spectroscopic imaging to address biological questions. And as James mentioned, I'm particularly interested in disease modeling in zebrafish, and I had previous experience working with infection models. So we started applying Raman spectroscopy for analysis, and we were working in a zebrafish model of tuberculosis, where we infect the fish with bacteria called Mycobacterium marinum, which is a close relative to the Mycobacterium tuberculosis, and it causes an infection that mimics the kind of early pathogenesis of tuberculosis inside the zebrafish. So in this work, we developed a workflow for identifying the bacteria in the fish and then compare the biomolecular profiles between different strains of Mycobacteria. So what we first observed was that when we infected the bacteria locally, they formed lesions and with dense clusters that could be quite easily detected using conventional histology with a Nielsen seal staining you can see here, which is the common way to visualize Mycobacteria in histological sections. But also these clusters of bacteria very clearly showed up when we performed Raman images. And further analysis revealed that these clusters contained very strong lipid contribution in the spectra. These are associated with peaks you can see that are annotated here on the right. And we identified these strong liquid contribution also in liquid culture bacteria. And looking into the literature, we saw that this had been reported before, and it's mostly caused by a very thick cell wall that all Mycobacteria has, and which also plays a very important role in the early pathogenesis. And having kind of identified this feature, we could use these lipid peaks to discriminate the bacteria from the surrounding zebrafish hosts. So we next use this to compare two types of infection, where we compare the wild type marinum to a mutant marinum, which contains a deletion in a locus, which codes for several important antigens that are involved in the early bacterial lesion formation. And we use the lipid peaks that I mentioned in the previous slide to extract the pixels containing bacteria. And we then compare the biomolecular profile of the wild type bacteria to those of the mutant called delta RD1 through a principal component analysis. And what we observed was that we could, through this analysis, separate the two groups quite clearly. And interesting, in our components, we observed that one of the main differences between the two groups of bacteria were the amount of saturated lipids that we found in this spectra. So this experiment demonstrates how we can use Raman spectroscopic imaging to extract biomolecular profiles from different bacterial strains during infection in zebrafish, and how they, this information can be used to discriminate and analyze the state of the bacteria during the infection. So moving on from this, we were also interested in developing imaging protocols to analyze living zebrafish. And for these experiments, we worked in a stab wound model, and then we tracked the wound response using Raman spectroscopic imaging over time. So the embryos were injured using a surgical needle, and this caused a localized injury in a single body segment or a somite, which you can see quite clearly when we stain the zebrafish muscle tissue with phalloidin. The embryos were then imaged using near-infrared lasers, which is the most biocompatible laser source that we can use for Raman. We then followed this wound response for 12 hours and recorded 3 successive images and analyzed the data using a multivariate analysis approach called Vertex component analysis. And interestingly, this analysis quite clearly could identify a differences in the Raman spectra between the healthy tissue and the wounded somite. In addition to that, we also identified a pigment end member stemming from the edge of the fish tissue. And we further compare the unwounded end member that this analysis revealed to the wounded end member simply by extracting the one from the other to look at differences in the spectra. And what we saw was that the unwounded end member contained a higher amount of proteins, while the wounded end member had a higher amount of especially lipids and also the carotenoid pigments that we saw especially on the surface of the fish in intact embryos. We then tracked these 2 components over time and saw a slight increase in the wounded component as this experiment progressed. So again, this demonstrates how Raman spectroscopic imaging can be applied to living embryo samples and used to track biological processes over time and provide relevant biomolecular information. And this is also one of the applications that we are really interested in developing further. So to conclude, we demonstrated both high resolution and 3D imaging that is applicable to zebrafish using confocal Raman spectroscopic imaging. And we have applied confocal Raman spectroscopic imaging for biomolecular profiling and discrimination of different mutant bacterial strains during infection. And we also demonstrated in vivo live imaging approach for zebrafish embryos and applied this for time-lapse analysis of a wound response. And for further information, I want to refer you to a paper that we recently published. James is also a co-author on this one. And it's in Nature Communications and was published, I think, just a month ago, more or less. Host of people to thank. A lot of people in the Stevens group, Molly, Connor, James, especially have and Mats have been contributing a lot to this project. Also for the zebrafish part, I had really good help in the Dalman group, which is based at Imperial College, and the Mostovi group, which is based at the London School of Hygiene and Tropical Medicine. And with that, I want to thank you all for your attention.
Thank you, Hakon. That was a fantastic talk, and we have plenty of time for questions. So if you have any questions, please post them in the group chat, and I'll relay them to Hakon. I'll get things started. So can you maybe just first of all talk a little bit about some of the limitations of Raman and maybe the comparison between using Raman and using more conventional fluorescence imaging? If I'm a biologist and I want to have a problem to assess, how would I decide which technique to use?
Yeah, I mean, there's several kind of important limitations to Raman. I think the two really important ones is the sensitivity and the time it takes to acquire an image. I mean, compared to fluorescence, I think that's kind of two completely different imaging modalities. I think most of your experiments, you want to start doing some kind of fluorescent imaging or histology, because in Raman, I think if you want to perform Raman on zebrafish, you really need to know what you're looking at. You need a very, very clear, hypothesis, I think, about the that you can investigate. I think the strength of Raman is that since you generate these biomolecular profiles which are loaded with information, you can kind of interrogate the metabolism or changes in the metabolism in really great detail. But I think you need to work within a kind of that type of questions. Like, that's why we've chosen like tumor metabolism, bacterial metabolism, and liver metabolism, because you need kind of a very well-defined question, I think. In terms of the time, typically these images takes hours, so that's also worth considering. You get a lot of information, but it's also quite time consuming to perform the experiments.
Okay, yeah, that makes perfect sense. And I think we have a follow-up question from Matthias, who says, nice paper, Hakon. For organ development tracking, I assume keeping the same zebrafish alive over longer periods of time is important. What are the challenges associated with this, and how suitable are standard optical microscopy setups if you want to combine these with Raman imaging?
You mean combine in terms of performing correlative imaging? Yeah, I can say a few things. I mean, one of the challenges with keeping a fish alive for a long time is actually that this is a growing embryo, and to image them, we embed them in agarose, which means that they start growing in the agarose. Some of the challenges with that is basically that the fish eventually will be crushed. So it can be tricky to keep the fish alive for a long time. There are maybe ways to do it, but I think with Raman it can be quite hard, because we are still imaging single planes, so we're quite sensitive to any change in the caused by the fish, say, moving around in the gel. In terms of correlative, I think that's a very, very interesting avenue to combine Raman imaging with fluorescent imaging. The one thing to keep in mind there is that in both cases you are detecting light, and fluorescence is by far the stronger phenomenon, which means that if you try to detect Raman spectra and fluorescent signal in the same wavelength, you will only see fluorescence. So background fluorescence is an issue actually in most Raman imaging. There are ways to deal with this. There is a paper from 2014, I think. Bennett is the first author, where they saw this by imaging fluorescent in the green region, and then moving the Raman laser further towards the red part of the spectrum. So there's no overlap, and I think it would be very, very interesting to see if someone could kind of combine our imaging capabilities here with a fluorescent setup, because I think that could be quite powerful. And referring to the question James asked in terms of or my answer to James, where I said you need a very clear hypothesis. Obviously, if you can identify bacteria with fluorescence and then just specifically image those areas, that would speed up your imaging and refine the analysis a lot. So I think that that's a super interesting thing, and it should be possible. It's not something we tried.
So with regards to that then, can you speak a little bit more about the resolution of the imaging, or if you wanted to focus on a particular bacteria, is that possible using this technique?
I don't think the resolution, the spatial resolution is the big limitation there. I think the limitation is the sensitivity. I think that this bacterial model worked very well because the bacteria grows in clusters, and that gives a very strong signal, which is easy to detect. The spatial resolution here is similar to what you have for a light microscope. So theoretically, it's down to 0.5 micron. But unless you have a very strong Raman signal, single bacteria, especially inside fish, I think we don't know this, but it's quite likely that there are single bacteria that we don't see in our study because they're simply buried in the rest of the zebrafish tissue. So I think that rather than the spatial resolution would be my concern.
Sure. Okay.
Yeah.
So I've one last question before we move on. So it's a more specific question. So you showed in your live imaging, you showed the progression of wounding, and you saw the wounded component increasing over time. Do you think that this could be applied to looking at the resolution of a wound site?
Possibly. I think the limitation we would have there is that the resolution is quite slow. So we would need to keep the fish then alive probably for a couple of days. I think that would be not an issue necessarily with the Raman system, but more with the embedding and by making sure we always image the same area of the fish. But yeah, that's definitely something that could be interesting to do as well. But yeah, like I said, the trick then would be to keep a growing embryo fairly fixed in place and alive for an extended period of time, which is a challenge.
Sure. Okay, thank you. That was fantastic. Thank you for answering the question so comprehensively. We will move on now to our second speaker. So Natalie, I'll hand over to you.